Welcome everyone. Today we are going to learn about ATP sensitive potassium channel. This channel is important because it helps in the secretion of insulin. Now I have written all the important points here that I will explain one by one. At first I want to mention that ATP regulates the closing and opening of this channel. Now when the concentration of ATP increases in the cell, then the channel closes, and when the concentration of ATP is lower, it actually remains open. So we know that ATP actually indicates the metabolic status in a cell. So in a way, we can say that this channel also can sense the metabolic status in a cell. So ATP regulates the closing. When the concentration of ATP is high, it closes, and when the concentration of ATP is lower in a cell, it actually remains open. Another important point is this channel is actually present in beta cells of pancreas. We know that from these beta cells of pancreas, insulin is secreted out. Third point, we have to remember that this potassium channel, ATP sensitive potassium channel, is actually a complex. It contains 4 alpha subunit and 4 beta subunit that makes a complex structure. Now the ATP subunit, the alpha subunit is actually the real potassium channel. We call it KIR. KIR means potassium inward rectifier 6.x. One subtype is present called 6.2. So KIR 6.2. So this alpha subunit is the real potassium channel, and the beta subunit is actually associated with this potassium channel. Now beta subunit is actually a ABC transporter. I have mentioned in my previous video, ABC transporter means ATP binding cassette proteins. Now in this case, this ABC transporter is called SUR. The full form of SUR is sulfonylurea receptor. Now sulfonylurea is a class of anti-diabetic drug. How this drug works, I will explain later in this video. So the beta subunit is a sulfonylurea receptor. Now 4 potassium channel subunit and 4 sulfonylurea subunit makes a complex structure. I will show the structure in this video.
This is the structure of ATP sensitive potassium channels alpha subunit and beta subunit. Now as I have mentioned, the alpha subunit is the real potassium channel called KIR 6.x. It contains 2 transmembrane alpha helix and 1 post segment, as I have mentioned in my previous video, ion selectivity. And this channel contains ATP binding domain where ATP binds. Now beta subunit is called sulfonylurea receptor. It also contains transmembrane domain 0, 1 and 2. These are all alpha helices. And it also contains a nucleotide binding fold 1 and nucleotide binding fold 2. And in this interface of this NBF1 and NBF2, magnesium ATP can bind. Now this is one example of 1 beta subunit and 1 alpha subunit. But in case of potassium, ATP sensitive potassium channels, this contains a complex contains 4 alpha subunits and 4 beta subunits, and it looks like this is the molecular structure of the whole KATP channels. Here 4 beta subunits and 4 alpha subunits makes the KATP channel complex. Now ATP, when binds in the ATP binding domain of alpha subunits, it closes the channel, and also in that situations, in the interface of NBF1 and 2, there is magnesium ATP. But when the magnesium ATP is converted into magnesium ADP, hydrolysis occur, this beta subunit actually overrides the inhibitions of ATP, and it actually opens the channel.
Now in general, when the cells is resting in condition, the potassium channel is actually remains open, and through these potassium channels actually potassium comes out. That is why it is called potassium efflux occurs. So through this channel potassium efflux occurs in resting conditions. Now in this insulin secretion mechanism, another whole channel is involved, that is called voltage gated calcium channels. We call V for voltage gated. So this channel can sense the voltage. Voltage means membrane potential of the cell. So here also another channel called calcium, voltage gated calcium channel is involved in these secretions. Ultimately, when calcium actually enters inside the cells, I will explain the total mechanism. When the calcium enters through the calcium channel, insulin containing vesicles present in the beta cells actually fuse and ultimately insulin secretions occur. Now when we take meal, the blood glucose concentration increases, and when blood glucose concentration increases, the glucose enters inside a cell through a unipotent GLUT protein. In case of beta cells of pancreas, this is called GLUT protein. Now when glucose enters inside the cell, it is metabolized by glycolysis, Krebs cycles, and ultimately by electron transport chain, it is converted into ATP. So when we take meals, blood glucose concentration increases, glucose enters, ATP generation occurs, and that increase in the ATP, actually what happens, this ATP binds with these potassium channels. Now basically in normal conditions, that means when we are not taking meal or when the concentration of glucose is actually lower in the blood, then this channels remains open in the beta cells, and through these open channels potassium efflux occurs. And we know when potassium efflux occurs, that actually makes a resting membrane potential, and the value of resting membrane potential is -70 millivolt. That means the cells is resting in conditions. Now when ATP concentration increases in the cell, then this ATP actually bind with these potassium channels. Okay, so when potassium channels attach with this ATP, it actually becomes closed. So it becomes closed. And now potassium cannot come out of the cells. So when potassium cannot come out, what will happen? More potassium will remain inside, and that will actually increase the membrane potential. So the membrane potential will rise from -70 millivolt up to 40 millivolt, and in that membrane potential, in that threshold, the closed voltage gated calcium channels, because at -70 millivolt the calcium channels remain closed, but when the potential increases by this closing of the potassium channels, then at this -40 millivolt the calcium actual channels open, and through this calcium channels calcium influx occur. Now calcium helps in the insulin containing vesicle to fuse and secretion of insulin. Now I have mentioned here that the beta subunit of this potassium channel is actually sulfonylurea receptor. Now sulfonylurea, this drug can target this receptor. When sulfonylurea bind in this sulfonylurea receptor, it actually helps in the closing of the channel. And as I have mentioned, when the channel closes, the membrane potential increases. This is called depolarization. So the cells become depolarized, and the voltage gated calcium channels open, calcium influx occur, that causes the fusion of this insulin containing vesicles and causes insulin secretions. So this is all about this potassium, ATP sensitive potassium channel. Now I want to mention two important points, that loss of function mutations of either this KR6.X or SU1 subunits of this channel complex can cause congenital hyperinsulinism, because loss of function causes these channels to remain closed, and in that closing state what will happen? Depolarization occurs and insulin secretion occurs. So higher amount of insulin hormone will be secreted out of these beta cells, and that causes congenital hyperinsulinism. Okay, and that can cause also hypoglycemia and life-threatening conditions. But the gain of function mutations of either this KR6.X or SU1 subunit of this channel can cause neonatal diabetes type 1, because here the channels actually has a reduced ATP sensitivity, and it actually becomes very hyperactive. That means it remains open most of the time, it do not close. So that's why the cell cannot secrete a good amount of insulin. So it can causes neonatal diabetes type 1. So hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching, and always keep learning.